Very good afternoon to all the EAPCET aspirants. This is your Prasanna ma'am from NEET WORLD. So, yes, in our previous class we have dealt with the complete framework of the chapter Cell the Unit of Life. So, today we are going to exercise the beginnings of this chapter, that is cell discoveries, cell theory, exceptions of cell theory, prokaryotic cell in detail, differences between prokaryotic cell and eukaryotic cell, and then we are going to look into in detail about plant cell and animal cell, after which we are going to learn about the cell wall. This is what we are going to learn today. Okay. So, here we go with the first and primary most statement from this chapter, that is cell is the basic structural and functional unit of life. What does this mean? Cell is the basic structural and functional unit of life. What does this mean? Okay. It is addressed as the basic structural and functional unit of life because a cell is independent in its structure or function. Okay. A cell is completely independent both in the structure and function. If you look into the hierarchy of organization in a living organism, first and primitive most things are what beta? Atoms. Okay. First and primitive most things include atoms, followed by group of atoms will combine to form a molecule. Group of molecules will combine to form a what? Micromolecule. Then group of micromolecules will form cell organelles. Cell organelles will be constituted inside the cell. A cell consists of what? Cell organelles, and group of cells which are similar in the structure, function and origin, they form what? A tissue. Tissues will organize to form an organ. Organs will organize to form organ systems. Group of organ system will make an organism. Okay, this will be the level of hierarchy. In this, the first independent primitive cell, both structurally and functionally, is what? Cell. Cell is independent both in the structure and function. Who identified the dead cell? Okay, so first it is Robert Hooke. Who has identified dead cells. How did he identify dead cells? He has identified from the bark of cork cells. Okay, from the cells of like cork cells, which is bark. Okay, bark or cork cells, one and the same. Okay, from here he has identified, he has observed the structures under the microscope as hollow cell like structures, which is just like honeycomb structures. Okay, which are just like honeycomb structures. As there is no protoplast inside the cells, it is hollow honeycomb like structure. Fine, right. These are addressed as what? Dead cells, as there is no protoplast. Then who identified the living cell? Fine, right. So, living cell is identified by second, Anton van Leeuwenhoek. Anton van Leeuwenhoek has identified the living cell. He has identified the living cells from the cheek cells of human, from spermatozoans or from single celled bacteria. Okay, from that he has identified the living cells. He is also the father of microscopy because he is the first person who has given out the lenses and identified the single celled organisms which are living. As such, who identified the living cell first? Anton van Leeuwenhoek. Here there is a question to be answered. Ma'am, from when this TS EAPCET BPC classes? It has started since a month, beta. Since a month it has started. Classes have begun since a month. Yes, you can follow these classes. Yes, you will definitely get, because see, I do not know your name exactly. You can please type out your name so that I can address you clearly. Okay, no issues. So, here in our classes the concepts are dealt from the basic level beta, from the basic level to advanced level, and at the end of each chapter you will be exposed to all the EAPCET previous year questions. Okay. So, this will be a really very useful session to you. You can watch the playlist or recorded sessions of the previous classes, because so far we have dealt with all the units, only cell biology and ecology are left from your class 11th text of botany, rest all chapters are done. You can go through the recorded sessions as well. Okay. So, here we are starting with cell biology. If you are joining live each day, then that will be helpful beta. Okay. Any doubts you are having, you can drop a message in the chat box, I will be addressing it. Okay. Yeah. Okay. So, next, who identified the nucleus? These three are the primary discoveries that you have to keep it in your mind. It is Robert Brown. Robert Brown has identified the nucleus in which cells? In the root cells of orchid. In root cells of orchid. Fine, right? So, these are the primary discoveries that you have to remember, who identified the dead cell, who has identified the living cell, and what did Robert Brown discovered. Okay, nucleus, and he has discovered the nucleus from root cells of orchid. Now, we will move on to cell theory. Okay. So, cell discoveries are done. Now, we will move on to cell theory. Who gave us cell theory? Cell theory is proposed by two major scientists. Okay. Cell theory is proposed by two major scientists. One is German botanist and the other is British zoologist. Okay. In the year 1838, German botanist whose name is Matthias Lydon he has proposed out one point by observing all the plant cells. He has just observed all the plant cells, and he said that all plant body is composed of cells and products of cells. Just he has observed all the plant cells and complete plant body. He has given out only one point, that is complete plant body, okay, is composed of cells and products of cells. Plant body is composed of cells and products of cells. Next, it is the scientist who is British zoologist. British zoologist in the year 1839. What did he propose? That he has proposed that he has observed both plant cells and animal cells, and he is the first scientist who has spoke about the limiting boundary. Okay, the limiting boundary of a plant cell is cell wall, is said by British zoologist in the year 1839. Remember this point, multiple times asked, okay, even in NEET and EAPCET. Okay, though he is a zoologist, he has said about the limiting boundary of plant cell. Limiting boundary of a plant cell is cell wall. Okay, limiting boundary of a plant cell is cell wall. Fine, right? Okay, limiting boundary of a plant cell is what ma? Cell wall over here. And he has also said out this point that all plant body or animal body is composed of cells and products of cells, and the limiting boundary of a plant cell is cell wall. Animal cell limiting boundary is cell membrane. Animal cell limiting boundary is cell membrane. Fine, right? Okay, these are the points that are dealt by what? German botanist, that is Matthias Lydon, and British zoologist, that is Theodore Schwann. Theodore Schwann. Okay, of course, cell theory all of you might be read even for EAPCET or for NEET or for your IPE exam. This is very basic. But then if you get statement based questions, you will be confused a bit again. So when you are getting statement based questions or assertion reason type of questions, you have to majorly remember that he has worked, Schleiden has worked only on the plant body, and he said that plant body is composed of cells and products of cells. Who spoke about the limiting boundaries? About limiting boundary of a plant cell or animal cell is being clearly dealt by whom? British zoologist Theodore Schwann. Remember these 2 points, okay. And then if we have to see the modifications, modified cell theory, once we will see. Nobody, either Schwann or Schleiden, has spoke about how new cells are existing. How new cells are formed is, nobody has dealt with that point. In the year 1855, it is the scientist Rudolf Virchow, okay, Rudolf Virchow has proposed a concept called as omnis cellula e cellulae, okay, omnis cellula e cellulae. Years are also important here, ma, okay, omnis cellula e cellulae. So under this, what did he say? Under this he has spoke that cells, all cells, new cells will arise from pre-existing cells. All the new cells will arise from what? Pre-existing cells. Clear, right? So these points you have to remember and highlight, after which you have to focus on the exceptions of cell theory. Before knowing the exceptions of cell theory, you should be clear on the note that cell theory is applicable to what type of cells. Cell theory is applicable to those cells which has single nucleus. Cell theory is applicable to, just a second, yeah. So cell theory is applicable to those cells which has cell membrane and single nucleus, which is single nucleus and cytoplasm. Fine, right? So nucleus along with cytoplasm is addressed as protoplasm beta. Protoplasm, which means what? Nucleus along with cytoplasm. Fine, right? And this should be encoded by what? Cell membrane. Okay. Only to these cells cell theory is applicable. Now we will speak about exceptions of cell theory, which means if more than one nucleus is there in a cell, to such cells cell theory is not applicable, and if nucleus is absent, to such a cell, cell theory is not applicable. To dead cells, cell theory is not applicable. Now we have to look into all the examples that are given in our book, which have binucleated, multinucleated condition and no nucleus at all. Okay. Now we will be looking into the exceptions of cell theory. Here we go. Okay. Here we go with exceptions of cell theory. Take down carefully, ma, very important, okay. Exceptions of cell theory. Fine, right. So first we will target enucleated cells. Cell theory is not applicable to enucleated cells. Enucleated cells include sieve tube cells, tracheids. Okay. To these cells, what? They are enucleated and cell theory is not applicable to them. And second is more than one nucleus condition, okay, which is multinucleated cells. If you can take the example of paramecium, paramecium is binucleated condition, right. So paramecium is one example where cell theory is not applicable to it. Rhizopus, which belongs to the class of Phycomycetes in fungi, which exhibits xenocytic condition or multinucleated condition, right, ma? So here one more example is what? rhizopus. Fine, right. So, to these cells again cell theory is not applicable. Okay. Again, here we go. So, to enucleated cells, to multinucleated cells, cell theory is not applicable. Okay. And then any more examples, ma? Opalina, which exhibits multinucleated condition. Clear, right? Take down these examples carefully. If you are having any doubt, just let me know. Okay. And the major exception over here is virus. Virus are known as nucleoprotein structures. Nucleoprotein structures. They do not have a nucleus encoded by a cell membrane. They just have what? A protein layer called as capsid. If we have to take bacteriophage as example, it just has a protein coat covering the genetic material inside. Inside the genetic material will be there, and genetic material is covered by a protein coat. There is no nucleus, no cytoplasm and no cell membrane. So, to all virus is the major exception of cell theory again. So, virus are known as nucleoprotein structures or infectious agents, or they are also known as living entities but not a living cell. Okay, they are also known as living entities but not a living cell. Clear, ma? Clear till here? Important beta. Okay, important. Fine, right? You have to know like what are the clear exceptions of cell theory. Very important. Clear till here? Any doubt anywhere? Yeah. Now we will move on to different shapes and sizes of cell. Again important, we may get match the following type of question from here. So, different shapes of cells, if you have to see RBC. Okay, RBC is round and biconcave whose diameter is around 7.0 micrometers in diameter. Okay, this is 7.0 micrometers in diameter. And white blood cells are usually shapeless, so they are amoeboid. Columnar epithelial cells are long and narrow cells. Nerve cell is branched and long, and nerve cell is the longest cell. Okay, nerve cell is the longest cell. Fine, right? And trachea is elongated, enucleated. Elongated and enucleated. And then we will look into mesophyll cells which are round and oval. Mesophyll cells which are round and oval. And then we see, ma, here we go with prokaryotic cell. Okay. So, prokaryotic cell, first we will look into its structure, then we will know about the gram staining. Okay. Unlike the plant cell whose outer boundary is a cell wall or an animal cell whose outer boundary is cell membrane, prokaryotic cell has an outer membrane called as cell envelope. Prokaryotic cell is encapsulated by what, beta? Prokaryotic cell is encapsulated by cell envelope. Fine, right? So, this cell envelope consists of 3 layers. Cell envelope consists of 3 layers. The outermost layer is known as glycocalyx. Fine, right? The outermost layer is called as glycocalyx, which has complex strands, okay, complex sugar molecules which are arranged like this. So, if they are loosely arranged, if the polysaccharides are loosely arranged, then the glycocalyx can be a capsule. Sorry, if they are loosely arranged, glycocalyx is addressed as slime layer. If it is tightly arranged, it is known as capsule. Fine, right? So, this is the outermost layer of the cell envelope. Second is what? Second is cell wall. Cell wall of a prokaryotic cell or a bacteria is made up of peptidoglycan, which consists of amino acids and polysaccharides. Okay, it consists of amino acids and polysaccharides. Take down carefully, ma. Fine, right. So, it has amino acids and polysaccharides, the cell wall peptidoglycan. And next is what? Cell membrane. Cell membrane is third innermost layer. Okay, innermost layer of cell envelope is cell membrane. Here we go. Okay, the third layer is what? Cell membrane here. Cell membrane. Cell membrane has some extensions or invaginations towards the cytoplasm. Okay, it has some extensions or invaginations towards the cytoplasm like this. So, these invaginations can be in the form of tubules, it can be in the form of lamellae, or it can be in the form of vesicles. These invaginations are known as mesosomes. Okay, mesosomes are very important beta. Mesosomes quite a times asked question. Hotspot you can say, hotspot segment of the prokaryotic cell you can say, wherein from mesosomes you got multiple questions, even in your NEET PYQ might be, even in your EAPCET might be. Okay, important. Okay, so here mesosomes are what? They can be in the lamellar structures, they can be in the form of lamellae, or they can be in the form of tubules, or they can be in the form of vesicles. Fine, right. Now, here these mesosomes, what will be their function? Structural description is clear to you, right? The outermost layer is what? Cell envelope. Cell envelope has 3 layers. Outermost is glycocalyx. If the polysaccharides are loosely arranged, if it is loosely arranged, it is known as slime layer. What is the benefit to that bacteria which has the outermost wall as slime layer, loosely arranged slime layer? The benefit is it will prevent the bacteria from loss of minerals and nutrients. So, in which type of bacteria you can see slime layer beta? Those bacteria which are present in the halophilic conditions or in the extreme salty or extreme acidic conditions, okay, wherein the slime layer will prevent what? Loss of water and minerals from the bacterial cell. It will prevent loss of water and minerals. Fine, right? Next, capsule. If the polysaccharide, that glycocalyx layer, if it is tough and thickly arranged, which is called as capsule, this, what is the benefit that bacteria will get if the outer wall layer is capsule? Okay, the benefit to the bacteria is it will prevent the bacteria from the attack of host immune system. Okay, it will prevent the bacteria, from host immune system. From host immune system. Fine, right? Any doubt still here? The slime layer and capsule is clear, right? So, capsule outer layer will be there for most of the pathogenic bacteria, and slime layer will be there for most of the bacteria which are exposed to thermoacidophilic conditions might be or halophilic conditions might be. And next cell wall is majorly peptidoglycan. It is composed of peptidoglycan, which includes the composition of amino acids and polysaccharides. Next is what? Cell membrane, innermost layer of the cell envelope, which has mesosomes. Okay, these mesosomes can be existed in the form of lamellae, tubules or vesicles. Now, here what is important? Functions of mesosomes is important. What are the functions of mesosomes? Okay, functions of mesosomes is highly important. first and foremost function is DNA replication. Okay. First, DNA replication or duplication. Second is cell wall formation of bacteria, cell wall formation. Third will be distribution to daughter cells. Distribution to daughter cells. And fourth is respiration. Fifth will be secretion. And sixth is increase in surface area. How mesosomes are performing all these functions? Just have a look. I will brief you very quickly. Just a second. Here we go, beta. First is DNA replication. The mesosomes which are present on two opposite sides will contract together and they will help in the DNA replication, cell wall formation and distribution to daughter cells. Okay. That is how these three functions are coordinated by the mesosomes which are present at the opposite ends. Okay. Next is respiration. There is a major statement that is mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell, because mesosomes here will be releasing out the respiratory enzymes into the cytoplasm. Okay. Mesosomes here will be releasing out what, beta? Respiratory enzymes. Okay. Mesosomes will be releasing out respiratory enzymes into the cytoplasm. Fine, right? So, that is how mesosomes of a prokaryotic cell are equivalent to mitochondria of eukaryotic cell. Major line, have a note of it. Okay. Mesosomes are equivalent to mitochondria. Fine, right? Mesosomes of a prokaryotic cell equivalent to mitochondria of eukaryotic cell. Clear, right? Next, secretion. It will secrete out the enzymes, okay, and some molecules. And as mesosomes are of these structures, lamellar and tubular structures, this will increase what? This will increase the surface area of the cell membrane. Okay. Mesosomes will increase the surface area of cell membrane. So, I hope complete cell wall is clear in your mind, from the outer wall layer to the inner wall layer. Okay. It is clear, right? Crystal clear, right? Take a screenshot of this and you can revise in that way as well. Okay. Take a screenshot and revise in that way as well, beta. Clear till here? Here we go. Next is about the cytoplasm. When you have to look into the cytoplasm of prokaryotic cell, cytoplasm is the main arena of all the cellular activities. What does this mean? Inside cytoplasm only all the cellular activities will take place. Yes or no? So, that is a major statement, just look into it. What is that? Cytoplasm is the main arena of all cellular activities. Fine, right? So, cytoplasm is the main arena of all cellular activities. This is clear, right? Yeah. Now, what does cytoplasm consist of? First is genetic material. Okay. First is what, ma? Genetic material. And next is organelles. All the organelles of a prokaryotic cell are non-membrane bound organelles. Prokaryotic cell does not consist of membrane bound organelles. So, it has non-membrane bound organelles. Just a second. Non-membrane bound organelles. So, here you see genetic material is also of two types. Genetic material is also of two types. Genetic material is also of two types. One is genomic DNA and the other is non-genomic DNA. Take down the flow chart clearly. In a single flow chart, everything you will get it. Take it clearly, ma. One is called as genomic DNA and the other is non-genomic DNA. First one is genomic DNA, or it is also known as nucleoid, or it is also known as chromosomal DNA. Highlight all the synonyms, we can expect any of these terms. Second is what? Non-chromosomal DNA, or you can say extra chromosomal DNA, non-genomic DNA or plasmid DNA. Non-genomic DNA or plasmid DNA. Fine, right? Two types of genetic materials. I hope you are clear on this note. Yes, right, ma? Here we go. First, about genomic DNA or nucleoid or chromosomal DNA. Genomic DNA consists of what? Genomic DNA consists of double-stranded circular DNA, and there are no histone proteins to bind that negatively charged DNA. It has polyamines other than histones. Other than histones, what does it consist of? Polyamines to hold that double-stranded circular DNA. Okay, and this genomic DNA will take care about all the metabolic activities that happens inside the bacteria. This takes care of all the metabolic activities that happens inside the bacteria. So, what is the first and foremost point? The composition. Composition is double-stranded circular DNA. Is there any positively charged proteins binded to it? Yes. Polyamines are present. Polyamines are present, but histone proteins are absent. Histone proteins are absent. Fine, right? Next is what, ma? It takes care about what? All metabolic activities of a cell. Takes care about all the metabolic activities. Fine, right? Next you see non-genomic or plasmid DNA or extra chromosomal DNA. Here if you see, this plasmid DNA has multiple effects on the bacteria. All bacteria, if you see of the same type also, based on the habitat the bacteria is living, the phenotypic characters will be varying. Okay. So, what plasmid DNA does is, it will give a specific phenotypic character to the bacteria, and plasmid DNA plays major role in resistance to antibiotics, and also plasmid DNA plays a major role in sexual recombination, that is conjugation. Here we go with that. So, first is, just a sec. First is what? It gives specific phenotypic character, to bacteria. Second foremost important point, it helps in conjugation. How is it helping in conjugation? Because plasmid DNA will have multiple factors. One such factor that plasmid DNA consists is fertility factor, which is F factor. So, those bacteria whose plasmid DNA consists of F factor are addressed as F plus strain. Okay. So, the F factor from the plasmid DNA is transmitted to F minus strain bacteria, okay, by from the plasmid DNA. From plasmid DNA, one F plus factor is transmitted to F minus strain bacteria plasmid DNA. So, plasmid DNA is also helpful in what ma? Conjugation from F plus strain to F minus strain. You will get more clear idea when I am dealing with sexual recombination of bacteria. Fine, right? As of now, you remember F factor is present in the plasmid DNA and it is transmitted to the F minus strain. Next is what? Third most important point is plasmid DNA will help in the resistance to antibiotics. Okay, this will be helpful in what? Resistance to antibiotics. Fine, right? This is the complete information about genetic material that is present in a prokaryotic cell. Now we will highlight moving on to the non-membrane bound organelles. Okay, what are the non-membrane bound organelles that are present in a bacteria? Fine, right? Non-membrane bound organelles. Here we go with non-membrane bound organelles. Yeah, look at here. Non-membrane bound organelles include ribosomes and second is inclusion bodies. First we will speak about ribosomes. Ribosomes, what type of ribosomes are present and where are they exactly present? Okay, ribosomes, if you see 70S type of ribosomes are present beta. Okay, all the ribosomes that are present are 70S type of ribosomes, and this ribosomes whose larger subunit is 50S and smaller subunit is 30S. Clear, right? Larger subunit of the ribosome is 50S and smaller subunit is 30S. These ribosomes are also addressed as Pallade particles, as these are discovered by George Pallade. Fine, right? So, how these ribosomes, where are they situated? Ribosomes can be present attaching to the cell membrane, or ribosomes can be present inside the cytoplasm. Okay, ribosomes can be present attaching to the cell membrane or inside the cytoplasm. And here in the prokaryotes, when we speak about ribosomes, we need to highlight one thing, that is polyribosome or polysome condition. Polysome or polyribosome condition. What is this polyribosome condition? During the time of transcription, if a single ribosome, if a single mRNA has from 5 prime to 3 prime at the time of transcription and translation, single mRNA, if it consists of multiple ribosomes like this, this condition is known as what? Polysome or polyribosome condition. And what does this result? This will result in multiple protein expression, which is known as polycistronic expression. Okay, this is known as what? Polycistronic expression. This will result in polycistronic expression. Clear till here? Any doubts anywhere? Now we will look into the second non-membrane bound organelle. Second non-membrane bound organelle is what ma? Inclusion bodies. What are these inclusion bodies? Here we go about inclusion bodies. Yeah. Second about inclusion bodies. These are the storage bodies that are present inside the cytoplasm of a prokaryotic cell. These storage bodies doesn't have any membrane, so you can address them as phosphate granules, synophysian granules or gas vacuoles also. Okay. So, these are storage bodies. You can address it as phosphate granules Synophycean granules, okay, or sulfate granules. Clear till here? Any doubt anywhere, ma? Clear, right? Yeah. Yeah, that's it. Okay, about the genetic material and non-membrane bound organelles that are present in a bacterial cell. So, complete picture is clear, right, about the cell envelope first, then about genetic material and about non-membrane bound organelles. Now, we will speak about motile and non-motile structures of a bacteria. Okay, what and where are the motile and non-motile structures present? Okay, motile and non-motile structures of bacteria. Fine, right, ma? Take the heading as motile and non-motile structures of bacteria. Here we go with motile and non-motile structures. First, motile structure here. Just a second. Yeah, motile structures you see. Motile, the only motile structure of the prokaryotic cell is flagella. Flagella, which is primitive, and it consists of, flagella consists of, if you consider this as a bacteria with 3 layers, and this is the outermost layer. Okay, first it originates from the cell membrane. Okay, remember it originates from cell membrane and it extends from the cell wall. Extensions are from cell wall and originates is from cell membrane. It has LPSM rings. Okay, first it has LPSM rings. Okay, this is called as what? Basal body. Basal body followed by hook followed by filament. Okay, basal body, hook and filament. Fine, right? And the microtubular arrangement inside, if you see, the microtubular arrangement is 9+0 arrangement. It is not as complex as that you will see in a eukaryotic flagella. And what is the protein that is present? Flagellin protein. In eukaryotic flagella, if you see, tubulin protein is present. Prokaryotic flagella consists of flagellin protein. Okay. Now, you see non-motile structures. Non-motile structures of a prokaryotic cell includes, non-motile structures includes pili and fimbriae. Okay. Non-motile structures includes pili and fimbriae. Pili has 2 functions. One is attachment to the substratum, and how you can describe the structure of pili. Pili structure is defined as tubular extensions which are extending from the cell wall. Tubular extensions of cell wall. Tubular extensions of cell wall. Fine, right? And if you take one pili which is connecting the 2 different strains of bacteria as we have discussed, one is F plus and one is F minus. So, this type of pili which is helping in the process of conjugation, that is sexual recombination, is known as what, ma? Sex pili, from which F plus strain is transmitted to the plasmid DNA of F minus strain. Fine, right? Clear? Now, so these are the 2 functions. One is attachment to the substratum, next is conjugation. How you can describe fimbriae as? Fimbriae is described as bristle like structures which are sprouting out of the cell wall. Okay. Fimbriae is described as what, ma? Bristle like structures sprouting out of cell wall. Fine, right? So, these are bristle like structures that are sprouting out of the cell wall. Fine, right? So, this is about Fimbriae, and this only function of Fimbriae is attachment to the substratum. Only function that it does is attachment to the substratum. Clear till here? Any doubt anywhere, ma, about motile and non-motile structures? Now we will see different types of bacteria based on the shapes. Okay, first is spherical shaped bacteria are addressed as what? Cocci. Okay, spherical shaped bacteria are cocci. Singular it is coccus. Okay, rod shaped bacteria, these are mostly pathogenic bacteria. Pathogenic bacteria are bacilli, and the singular form is bacillus. Next is what? Spiral shaped, spirillum is singular and spirilla is plural. Next, vibrio and vibrium, these are what? Comma shaped bacteria. These are comma shaped, these are spiral shaped. Fine, right? Now we will see different sizes of cells. If you see a typical bacteria, it is of 1 to 2 micrometers. See, shapes and sizes of cells are very important. We can expect a match the following type of question. Have an additional focus when you are looking into any kind of diagram of text. Next is PPLO, pneumonia like organism, which is just 0.1 micrometers. Viruses can be from 0.02 to 0.2 micrometer in diameter, and a eukaryotic cell is as large as like 10 to 20 micrometers in diameter. Till here everything is clear, na beta? And here if you see a mycoplasma, which is the smallest cell, has how much diameter? It is 0.3 micrometers in diameter. Mycoplasma is the smallest cell, which is just 0.3 micrometers in diameter. And then if you see ostrich egg of ostrich is the largest cell. And what is the longest cell? From shapes we have learnt. Nerve cell is the longest cell. Largest, smallest, longest, keep it in your mind. Okay. So, this is about sizes of cells. Now, we will look into Gram staining process. What is Gram staining? Who gave us Gram staining? When we have dealt about the second layer of cell envelope. Okay. Second layer is what? Cell wall. Based on the composition of cell wall and based on the thickness of cell wall, we have two different types of bacteria, that is gram positive bacteria and gram negative bacteria. This is classified by the scientist Christian Gram, who has given out Gram staining. Okay. This is by the scientist Christian Gram. Fine. He has identified Gram staining. What did he do? He has taken two different bacteria in two different petri dishes, and then first he has stained it with crystal violet color. Okay. So, cell wall, it has peptidoglycan, right, ma? It has what? Both proteins and sugar molecules, and those cell wall which consists of high amount of peptidoglycan, like 80% of peptidoglycan, will observe the crystal violet color. It will observe the crystal violet stain too much, and the bacterial type which has less peptidoglycan, whose peptidoglycan layer is just of 20%, it will not absorb the crystal violet stain. So, what happens? Two strains, one will absorb the crystal violet, one will not, and then they are treated with a mordant, that is iodine, and then after decolorization, both the bacterial stains are again stained with, both the bacterial strains are again stained with safranin. Okay. So, the one which has absorbed crystal violet, it will not give the other color, it will retain the stain of violet, it will retain the stain of violet, whereas the other strain which has not absorbed crystal violet, it will give you pinkish color as it has absorbed safranin, it will not retain the crystal violet color. Okay, here it will not retain crystal violet, and here it retains the color. Fine, right? Gram positive and gram negative. As such, the one which retains the color is gram positive bacteria, because gram positive bacteria has 80% of peptidoglycan, which is thick in composition of amino acids and polysaccharides. It will absorb the strain. Okay. So, gram positive bacteria has what ma? It has 80% of peptidoglycan, and it consists of murin. Okay. It consists of murin, it consists of 80% of peptidoglycan. Fine, right? And gram negative, if you see, gram negative bacteria, it just has 20% of peptidoglycan and murin is absent, rather it consists of what? Pseudomurin. Here you can easily make out from the diagram gram positive and gram negative bacteria. If you have to take the examples of gram positive, you can take Bacillus, Clostridium, Corynebacterium. Gram negative, if you have to take the examples, E. coli is the major example, Acetobacter, Azotobacter, Salmonella. These are the examples of what? Gram negative bacteria. Till here everything is clear, right beta? Gram positive and gram negative. Any doubts anywhere? Yeah. If till here it is clear, we will wind up the session. Okay. So, in today's session, we have completely fixed out the prokaryotic cell. In our next class, we will be looking into the differences between eukaryotic and prokaryotic, and then we will look into the eukaryotic cell in detail about the cell organelles of a plant cell, cell organelles of an animal cell. Then we will move on to cell wall and cell membrane in detail, okay, in the coming class. Okay. Till then, exercise today's class. If you are having any doubts, you can drop a message in the comment box. I will be addressing all your doubts. Okay. So, that is all for today's session beta. Thank you.